replication of DNA or duplication of DNA or doubling of DNA. One DNA molecule that produces two DNA molecules. And this process called autocatalysis. Replication of DNA or duplication of DNA or doubling of DNA also called autocatalysis. Here, DNA that shows the two properties, autocatalysis and heterocatalysis. In case of autocatalysis, from one DNA, two DNA molecules are formed. In case of heterocatalysis, from DNA, RNA is formed. This replication of DNA, first observed by and expressed by Watson and Crick. Replication of DNA first proposed by Watson and Crick. And this replication of DNA is semi-conservative method. Semi-conservative method of replication of DNA first proposed by Watson and Crick. They proposed the double helical model of DNA. At the same time, they also proposed semi-conservative method of DNA replication. What is the meaning of the semi-conserve product? One DNA, suppose it is the parental generation. During its replication, the two strands are separate from one another. It is mother strand, it is mother strand, mother strand, mother strand. The two mother strands are separated. New strand is formed. So, two DNA molecules are formed in replication. Out of the each DNA molecule, out of the two strands of DNA molecule, one strand is parental and one strand is newly synthesized. That means half of the parental DNA conserved, protected, and another half is newly synthesized. So that method is called half conserved, protected method. This method, semi-conservative method of DNA replication, first proposed by Watson and Crick. Experimental proof of replication of DNA. Next one, experimental proof of replication of DNA. This replication of DNA generally occurs in S phase of interphase in cell cycle. Replication of DNA, duplication of DNA, or doubling of DNA, or semi-conservative method of DNA occurs in S phase of interphase in cell cycle. DNA replicates semi-conservatively, experimentally proved first by Messelson and Stahl.